Hello everyone, welcome to your favorite learning channel EduRev. In this session, we are going to learn about an interesting experimentation which explains the base of our genetics. That is, inheritance of one gene from chapter Principles of Inheritance and Variation. So, let us begin with our topic. Before jumping on to big terminologies and experiments, we must know what are chromosomes, what are genes, and what are alleles, as they are the basic foundation of our genetics. So the image in front of you is the image of chromosomes that you see. These are the chromosomes. And from the image itself, you can see that chromosomes always occurs in pairs. It is a very, very important point that chromosomes always occurs in pairs. Now what do we know about chromosomes? That they have double-stranded DNA in it. They have double-stranded DNA in them. Yes? Now what all are present inside the DNA of humans? Now for example, you see a person and you have to describe that person. Now how will you describe that person? Either you will describe that person's height, or you will describe that person's hair color. Yes, either you will describe the person of a hair color. Now these two traits of a human, either you talk about the height or the hair color. Now these are encoded in a particular section of a DNA. They are encoded in a particular section of a DNA which are called as genes. So now we can define a gene as a gene is a section of DNA that encodes for a certain trait. Certain trait, I am repeating it again, that either it will encode for height or it will encode for the hair color. Now, if you describe the same person again, if a person asks what was that person's height, either you will tell that person was tall or you will say that person was dwarf. And if asked about his or her hair color, then you might say that the person's hair was black, or you can say that person's hair color was brown. Now what are these characters? And you also see that the traits that you just saw are contrasting from each other, means different. Either that person will be tall or that person will be dwarf. So these contrasting pairs, traits which are present on a particular gene, are called as alleles. Now these are called as alleles. So we can define an allele is a variant form of a gene which contributes in the diversity in phenotypic expression. Phenotypic expression means characters which are visible, that you can see from your own eyes. You don't have to study that person's DNA to know what kind of trait that person has, because it's very clearly visible. Either that person will be tall or dwarf, or that person's hair color might be black or brown. So these contrasting traits are called as alleles which are present on a particular gene. Yes, which are present on a particular gene in the section of DNA. Let us touch another important topic briefly for your better understanding of what we are going to learn. And the topic is gamete formation. Now the image that you can see on your screen is the image showing process of gamete formation. Now, do you know how many chromosomes are present in humans? So the number of chromosomes that are present in humans are 46. Now, till now we know that when an offspring is produced, that offspring has half chromosomes from its mother and half from its father. Let me show it with the help of a simple diagram. Now, as we know that chromosomes always occur in pairs. So, this chromosome is the female chromosome. Now, she is female. Yes? And Another chromosome is of male. Now both have 46 number of chromosomes, 46 in female and 46 in male. Now when the sexual reproduction takes place, the number always reduces both in male and female to 23. Means it reduces to half number of chromosomes, half in female and half in male. Then the half of 46 is 23. Yes, 23. Now when sexual reproduction takes place, both chromosomes from female and male gets combined to form an offspring, a baby. Yes. Now which has now 46 number of chromosomes in it. 23 from female and 23 from male, forming a 46 number of chromosomes, which is the actual original number in case of humans. Now with this we know that gametes are formed from reductional division. Yes, in this image also you see that this original chromosome number is getting reduced. It is getting reduced here to form finally 4 gametes, and these all 4 gametes now have 23 number of chromosomes each. 23 here, 23 here and 23 here. Now similarly, when the sexual reproduction is going to take place, from these 4 gametes, any of the gametes will go and fuse with the female gamete, forming an offspring. Coming to our topic now, which is inheritance of one gene. Now in this we study various symbols and terminologies which are very important because the whole understanding of topic depends on it. So the first one are the homozygous chromosomes. Now what are homozygous chromosomes? An individual having 2 identical alleles, identical, same. That is, either that chromosome will have BB, capital B, or small b, as you can see in this image. So now this type of chromosome having 2 identical alleles on the same location in chromosomes are the homozygous chromosome. Now next one is heterozygous. Now heterozygous chromosomes are when an individual have 2 different alleles, that is, either the chromosome will have capital B or small b in the same location. Now these 2 are different. Capital B and small b on the same chromosome are the different alleles, the different types of alleles which are present, and that is why this chromosome is known as heterozygous chromosome. Next terminology is the phenotype. Now phenotype is the physical appearance of the trait, that either that person will be tall or dwarf. So these are some visible characters that you can see from your own eyes if a person is tall or if a person is dwarf. Now next is the genotype. Now genotype is the genetic makeup. This is the genetic makeup of an individual, that if a person is tall, so the genetic makeup for that person will be capital T. And if a person is dwarf, then the genetic makeup for that person will be small t. So these are the genetic makeup of a person. Next are the dominant traits. Now these are the traits which are always expressed when the connected allele is dominant, So when you look at the height of the world population, so maximum people, you will find out that maximum people are tall. Yes. So we can say that tallness is the dominant trait in the humans. Yes. Now, if you talk about the genetic makeup, how will you know? Now they have said if the connected allele is dominant, means capital T. The T we are taking as a symbol of tallness, and as we know that genes or alleles always occur in pairs, so we see that capital T in pair resulting in tallness in humans. Now they have also said that it also occurs even if only one copy of the dominant trait exists. That is capital T. Means the another possibility is if the allele has one capital T and one small t. Now in this case the person will again be tall. And how? Because this capital T is dominating the small t. It is not letting that small t to express itself because this capital T has dominant features. That is why that person will again be tall. So this is known as dominant traits. Now in case of eye color also, brown color is also dominant. Brown eye color is dominant. Now next one is the recessive trait. Now what are recessive traits? Recessive traits are obviously contrasting the dominant traits, and they are expressed only if both the connected alleles are recessive. Means both the alleles should have small t. Then only that person will be dwarf. Yes? Unlike the dominant traits in which we have seen that if the allele are capital T small t also, then also a person will be tall. But in case of recessive, only small t should be there for a person to become dwarf. Now in case of eye color, the blue eye color is recessive. So we have seen two traits. We have talked about tallness and about the eye color. Let me tell you a story about Gregor John Mendel. Now he is the person because of which we are studying this topic. So Gregor John Mendel was born in Austria and he was very intelligent in his studies. But he did not like the pattern of society, so instead he became a monk in a monastery. Now in the monastery, he taught students various subjects, and because he had nothing to do apart from teaching the students, so he started growing garden peas, whose botanical name is Pisum sativum, in the garden of his monastery. So yes, he knew about the terminology about heredity. He knew a little knowledge. He had a little knowledge about heredity, in which he knew that when an offspring is produced, that offspring has the intermediate characters of both male and female. Yes, when an offspring is produced, male, female, that offspring has intermediate characteristics. So, but when he observed the peas that he grew in his garden, he saw that the height of a pea was different. Some were very tall, some were dwarf, and some were intermediate. And also when he looked at the pods of peas, he found that some peas were round, some were wrinkled. So he got very curious looking at that, and he didn't knew about why it is happening. So he started experimenting, and he experimented on the garden pea for about 7 years, that is from year 1856 and 1863. Now the name of years are very important. It is also given in your NCERT, so you must remember the dates. So he started experimenting on garden peas. He experimented on garden peas because they were quite easy to grow, and also getting the data out of it was quite easy for him. Now later on, according to his observations, he later on gave 3 laws of inheritance based on his observations, and finally published his work in year 1865. But obviously no one believed his work back then. But after several years, his work got republished, and then people believed that what Gregor John Mendel has given the experimentations, it was all true and was the basis of the genetics. Mendel in his experimentation observed some contrasting traits in garden peas, and those were the 7 contrasting traits that he observed. And contrasting traits, as we know, are either they are the dominant ones or the recessive ones. So the 7 contrasting traits that he observed in the garden peas were the shape of seeds, the color of seeds, the color of pods, shape of pods, plant height, position of the flowers, and the flower colors. So these were the 7 contrasting traits and are very important from the perspective of NEET and your boards as well. So these contrasting traits were, in case of shape of seeds, the round shape of seeds were dominant and wrinkled one were the recessive. The color of seeds, yellow, was dominant and the green color was recessive. Now I'll write down D and R. D is for dominant, R is for recessive. Now the green color pods were dominant and yellow color were recessive. Now shape of pods, if full, was dominant and flat and constricted were recessive. Plant height, the tall ones were dominant and the short one were recessive. Now the flowers at the leaf junction was dominant and flowers at the tips of branches were recessive. The flower color purple was dominant and flower color white was recessive. Now the question arises is, why a particular gene is dominant or recessive? For example, these are the pair of chromosomes which are having genes of height, let's say. So here is a particular section of a gene in the DNA. Here's a particular gene in a DNA. Now these genes are present on the same location on 2 chromosomes, and these are encoding for a trait of a plant that is for height. So I will write down these are encoding for height. So as we know that genes are present in a section of DNA, Now this DNA after transcription produces mRNA. It produces mRNA, and after the process of translation, mRNA produces proteins. These are produces proteins. Now, if the gene encodes for the height, if this gene is encoding for height, then the proteins which are produced in this case are giving height to the plant. So I will write down, it is because of these proteins which are produced from this section of DNA, these are responsible for the tallness of a plant. Yes. Now the same thing is happening in the another chromosome as well. So here is the DNA section of DNA, which is transcribes to mRNA, which again translates into the proteins, which is getting translated into the proteins. So this is also responsible for giving height. So I would say, or I would rather write down the symbol T for tallness, capital T and capital T, because both the chromosomes and both the genes of the chromosomes are helping in producing enough proteins which is making the plant tall. Now what if this second chromosome has a faulty DNA, or if mutation occurs in this case? So if this DNA is faulty, then it will produce a faulty mRNA also and faulty proteins as well. Means it is not producing enough proteins now. So I won't write down tall here now. I would rather write down that it will give dwarfness to the plant because it is not producing enough proteins. It is not producing enough proteins. So that is why it is encoding for dwarfness now. So I won't write down capital T here, but instead I will write down small t, which is for the recessive gene. But in this case also, the plant will be tall only. Why? Because the proteins produced by the first chromosome or the left chromosome, it is producing enough proteins for plant to become tall. So this means This capital T is dominating the small t. It is dominating the small t, and that is why the plant remains tall. That is why the plant will remain tall. Now, since the genetics is all about probability, and then keeping that in mind, there are some cases as well where both the genes of chromosomes are faulty. It means the DNA of this chromosome will be faulty. So it will produce a faulty mRNA, which produces a faulty proteins. It means now both the chromosomes, both the genes in the chromosomes are not able to produce enough proteins, so it will also encode for dwarfness. So now the symbol will change into recessive gene, and it will also become small t. Now these both are not dominating each other, and if they are not dominating on each other, that means they are expressing themselves as recessive genes which will encode for dwarfness of a plant, and the plant will become dwarf in this case. So we see that if for a plant to become dwarf, both the genes should be recessive. Both genes should be recessive for a plant to become dwarf. Having learned some basics of genetics and how dominant and recessive genes are formed, we will now explore the experimentation given by Gregor John Mendel, which is the inheritance of one gene, our topic, which is also known as the monohybrid cross. So monohybrid cross is a cross between two types of plants of same species, considering only the transmission of one character. It means we are going to study about the cross between two types of a plant of same species. As you can see in this picture also, that the plants shown in this image are of same species, but here we are considering the transmission of only one character, that is the character of height. So as you can see, either the plant will be tall or the plant will be short or dwarf. So let us now study this experimentation in detail. Let us now begin understanding the monohybrid cross. In the first look, it will look a little bit tough, but it is actually not. It is very easy. So let us begin. As we know that Mendel has taken the one character for monohybrid cross, that was height. So we will talk about height in the monohybrid cross now. So as we know that he has crossed a tall plant with a dwarf plant, but here they have written pure tall. Now what do you mean by pure tall? It means the two genes which are present in the tallness or the tall plant are the capital T, capital T. That is why they have written it as the pure tall, or it is also called as the homozygous tall because the genes are same. So he has crossed the homozygous tall with the pure dwarf having small t. So by studying gamete formation, we know that the only one gamete from male and female will get transferred into the offspring. That one T is coming from one parent and another T is coming from another parent. So this plant is now tall in the first generation, or called as F1 generation. In the image also they have mentioned F1 generation or the first generation, in which we get hybrid tall or called as heterozygous tall. And why we are saying it as heterozygous tall? Because it has two different genes, capital T, small t. Yes, different in terms of symbol. So that is why it is called as heterozygous tall. And also we know that this capital T will dominate the small t, and the reason we have discussed in the previous topic. Now, after crossing it for several years, he found out that the first generation plants are always tall. They are always tall because of how this capital T is dominating the small t. So he did one thing. Then he crossed the two plants of F1 generation only. So he crossed two plants for F1 generation only, in which these two plants are tall. So these are the plants from F1 generation. Yes. Why? Because these are the heterozygous tall. So after crossing them, he saw that some plants were tall. Yes. But one plant came dwarf. So he got surprised that how come one plant is becoming dwarf and others are tall? Because I have crossed two tall plants. So he used some statistical formulation in the form of Punnett square. So the square you see here is the Punnett square, which Mendel used to know that what is the genetic makeup of the plant that I have crossed. So he made a square, which is the Punnett square, which he divided into several blocks. Yes. So this is the block he made. Now, male plant, he took a male plant, capital T, small t. He divided these genes into each block, capital T, small t. Now, after combining or crossing it, so he found out if I'll cross this capital T with capital T, the genetic combination will become capital T, capital T. Now, if I'll again cross this capital T with this small t, then the genetic makeup will become capital T, small t. And if I am crossing this capital T with this small t, then again the genetic makeup will become capital T and small t. And if I'll cross this small t with small t, then it will become small t, small t. So this you can also find it in this Punnett square, also in the image. So he saw phenotypically, visibly, that there were three plants which were tall, 1, 2 and 3. He saw that these plants are tall and one plant has become dwarf. So phenotypic ratio. So the ratio he deduced from his observation, visible observation, was the ratio was 3:1. Phenotypic ratio, phenotype was 3:1, in which 3 plants were tall and 1 plant was dwarf. But when he tried deducing the genotype or the genotypic ratio of the F2 generation, this is the F2 generation. This is the F2 generation. When he tried deducing the genetic makeup here, he saw that one plant was homozygous tall. Homozygous tall, I will write 1 here. And 2 plants were heterozygous tall. Hetero, with different genetic makeup. 2 plants were heterozygous tall and 1 plant was altogether recessive or dwarf, having small t. As I have told you that a plant to become dwarf, both the genes should be recessive, and in case of dwarf, you can see that both the genes are recessive. So with this, he got his phenotypic ratio and genotypic ratio as in phenotype 3 tall, 1 dwarf, and in genotype 1 homozygous tall, 2 heterozygous tall, and 1 dwarf. So this cross is very important. Knowing its phenotypic and genotypic ratio is very, very important from the perspectives of your boards and NEET both. See, visibly, one cannot know if a plant is homozygous tall or a plant is heterozygous tall without knowing its genotype. It is not possible just by looking at it. That is where the test cross comes into the light. So, test cross is an experimental cross of an individual organism of dominant genotype but unknown genotype, dominant phenotype but unknown genotype, and an organism with a homozygous recessive genotype. You don't have to get confused looking at this definition because we are going to break it down. And how? That crossing an individual with dominant phenotype. Now, dominant phenotype of a plant, for example, let us say, take the height, capital T, capital T. So, this is the dominant phenotype because you will find that the plant is tall. But you don't know its genotype yet. We don't know if it is homozygous tall and if it is a heterozygous tall. So, I am just going by this capital T first. And then I am crossing it with an organism with a homozygous recessive genotype. It means the cross should always be with recessive genotype only. Means the test cross will always be with the dwarf plant of any dominant phenotype we are talking about. So here the result will be one capital T from one parent and one small t is coming from another parent. So now the plant here will be tall. And another case is, if I am taking a dominant phenotype will be tall again without knowing its without knowing its genotype, and I am again crossing it with the dwarf plant. Again I am crossing with the dwarf plant. Now here I have to draw a Punnett square. Here I will draw a Punnett square like this, in which I am going to make this cross. Now here is one parent capital T small t and another dwarf parent small t. Now if I'll cross it, if I'll cross this t with this t, then capital T small t, these two small t's, small t, the combination will become small t. And if I'm crossing the small t with the capital T, then here it will be capital T small t. And if I'm crossing it again with small t, then the genetic makeup will be small t. Now in this case, you see that two plants are tall. The two plants here are tall. And two plants are coming dwarf. So here now the ratio is 50-50. Half of the plants are coming tall and half of the plant are coming dwarf. Now, why do we do test cross? Till now we have explained everything, that what happens when a dominant phenotype is crossed with a recessive genotype. So basically the function of test cross is to determine whether an organism which is dominant is homozygous or heterozygous. If the plant is homozygous, or if the plant is heterozygous, because we don't know just by looking at it. So that is why we have to do the test cross, in which we are crossing a parent plant with a dwarf plant, recessive plant. So with this we know that in the first case, if we are crossing it with the homozygous tall, if I am crossing it with the homozygous tall, which I don't know yet, and if the F1 generation is all tall, then it is very clear that the tall plant by which I have crossed this dwarf plant is homozygous tall. And in the second case, if I am crossing again with a tall plant which is genotype I don't know yet, and if I am crossing with a dwarf plant, and if in my F1 generation, if I find that a dwarf plant is also coming, it means my tall plant, my heterozygous tall plant had one recessive gene, one small t my tall plant had, because of which the dwarf plant has now appeared in F1 generation. So you now know the function that it is done to determine if the organism which is dominant is homozygous or heterozygous. Yes. So now with this, our today's topic is done. I hope you people have understood about the monohybrid cross and test cross clearly. You can now go to EduRev and attempt its test to understand this chapter in depth. You can unlock all the logged videos, docs and tests of class 12th with EduRev Infinity Plan and ace your exams at less than 80 rupees per month. Thank you.